All right, guys, so in this video, we're going to be going over gene expression, and in this first video, we're going to be going over prokaryotic gene expression. So before we get too far into it, I just want to remind you guys of what that word gene expression means. And again, that's just really saying you're controlling of protein synthesis. So you just got to understand that it's really important for cells to know when to make a protein and when not to. So gene expression just means control of protein synthesis. So that's going to be the focus on this chapter. Let's kind of get into it. And before we do that, I always like to kind of at least recap what is the basic structure of a prokaryotic cell. So the big things that you want to remember is that there is no nucleus. They have what's considered a nucleoid region. And their DNA is circular. So all of this right here in the center is this big, huge circular chromosome. And they also have little tiny circles of DNA called plasmids. So there's little tiny circles of DNA that are going to be referred to as plasmids. But regardless if it's the big circular chromosome or the plasmid, they both do protein synthesis. So you really want to make sure that protein synthesis can occur based off of the large chromosome as well as the little plasmids. So other mentionable features that these prokaryotic cells have that the AP test likes to sneak on you guys is that they have a cell wall. So remember, cell walls are going to be in plant cells, but also in these prokaryotic cells. And we did a lab earlier in the year where we stained it, the gram positive, gram negative. So the cell wall is also there. Now, other than that, let's kind of get into a nice revisitation of what happens with these bacteria cells or prokaryotic cells is that they replicate very, very quickly, and they replicate via binary fission. So they are considered asexual. So basically what that means is no mitosis. So when we say that they're asexual, that means that they replicate just by doing not mitosis, just we call it binary fission. No mitosis, they replicate, and it's pretty quick, so every 15 minutes or so. So one thing that we got to remember is how do these bacteria cells, if they don't do meiosis, if they don't do sexual reproduction, how do they get diversity? And it really comes from a couple of ways. One is mutations. So just like all living things, mutations increase diversity because they introduce a new phenotype. But also, in terms of genetic recombination, how do they actually get new DNA sources? And that can come from a number of ways. It can come from other bacteria, literally dead bacteria can actually, DNA can enter the cell. It can be transformed as well as shared during transduction. So basically genetic recombination, the question that you want to make sure you're able to answer is how do prokaryotes increase diversity? So how do they get these new strains of bacteria? How do we get these new strains? Why do they keep showing up? So the big three are going to be transformation, transduction, conjugation. I'll briefly mention transposable elements at the end. But transformation, transduction, conjugation, all of these are going to be ways to increase diversity. So they're pretty easy. So let's just kind of make sure you feel okay on what's going on with these. So the first one is going to be called transformation. And as the name implies, you are creating recombinant DNA, which means you're creating a new type of DNA in the bacteria by inserting DNA fragments from what are considered donor cells. Now, that DNA can come from a human. It can come from, in a lab setting, we do bacteria transformation all the time. But these DNA fragments can come from dead bacteria, as a couple of studies have shown that even if the bacteria is killed and they insert the gene, bacteria can pick it up. So anytime bacteria picks up new DNA, we're going to call it transformation. We can actually transform it. So this is showing you the overview. Remember, it's the chromosome, the big DNA. It has a couple of genes, A, B, the A gene, the B gene, the C, the D, the E. But if it inherited these from a random source, we're going to call it transformation. And when it says remember the Griffith experiment, that's just saying when they took a dead rat, they injected one rat strain with a live bacteria culture that was dangerous, and they killed the rat. They also injected the same bacteria, but they killed it, but they introduced that dead bacteria into the rats, and they died as well. And that just showed that the bacteria picked up that DNA fragment. So the next one is going to be transduction. So how else can bacteria get their genetic information changed? and that's involving bacteriophages. Now, if you don't remember, these bacteriophages are going to be a virus. So this terrifying looking thing is going to be called a virus. Specifically, it's called a bacteriophage. So a bacteriophage is a virus that directly infects bacteria. So hence the name bacteriophage. So we also call these things phage for short. And so all this is it has DNA. This phage docks on the bacteria. It injects its DNA. It uses all of the bacteria cell's machinery to make copies of itself. And what that does is then as these cells, or I should say, as these viruses make more copies of itself, meaning the virus makes more of its protein code, it makes more of its genetic information, it's showing you that the cells incorporate their phage DNA. Basically, the virus replicates so many times that the donor, or I should say the host bacteria cell, often bursts. Now, we get into the lytic and lysogenic cycle when we get to chapter 19, but just for now, understand that transduction involves a virus. Virus injects its DNA into bacteria. Now, the last one is probably the most common, as well as one of the more craziest things, in my opinion, that bacteria can do. When we think of bacteria cells conjugating, so this is showing you that it has this plasmid. So the plus sign means that it has the plasmid. Now, if bacteria cells come in contact with each other close enough, they can exchange and make a copy of that plasmid and share that plasmid with a nearby bacteria cell. Well, why does this matter? Well, maybe this plasmid has the gene that makes it resistant to an antibiotic, and it can share that resistance DNA with a nearby bacteria cell. So what normally did not have the plasmid, it can receive the plasmid from nearby bacteria. So those are kind of the three main key players on how bacteria can actually change their genetic information. Transposable elements is often referred to as jumping genes. So genes can actually move from one location to another. And basically, the point for this is that it's going to turn on protein synthesis typically when this happens. So we haven't talked a whole lot about operons yet, but basically if you remove a gene and you insert it somewhere else, it makes it resist several antibiotics, and it can become drug resistant. So it's often referred to as genetic shuffling. So that's really the only thing that you're going to have to, if you ever get a question on that, it's about turning on protein synthesis that normally should be off. So the focus on this video is going to be about how is gene expression going to be controlled in these prokaryotic cells. So as simple as these cells are, they have to be able to control protein synthesis in a way that makes sense. So we're focused on gene expression. How do these little guys turn on and turn off protein synthesis? And that stems from what's called an operon, okay? Now there's two types. We have inducible operon and repressible operons. Your job is to be able to identify if it's inducible or repressible, okay? So all of this is going to be regulated by feedback inhibition. So how do we turn on or turn off enzymes? So let's kind of get into this. There's two ways that these things can regulate their gene expression, okay? Feedback inhibition, which is when enough product is made, the whole system shuts down. So basically, if you look here, here's your precursor, right? You have your starting molecule. It takes an enzyme, and then you eventually get your last product, which is tryptophan. Tryptophan can bind allosterically to this first enzyme, and it shuts the whole thing off, right? So when we talk about feedback inhibition, the way I like to think about it is you turn off the enzymes. So basically what this pathway in purple is showing you that, okay, if we made enough tryptophan, if that's our product, we can actually turn off the enzymes. Now, what is the difference between that and this form of feedback inhibition of gene expression, right? So let's say you make enough tryptophan. Tryptophan is going to turn off the enzymes that have already been made, but also tryptophan is going to stop the production of the enzymes to help make it. So there's a difference. Feedback inhibition turns off enzymes that have already been made. Gene expression is going to turn off the production of the enzymes. So you got to, and when you think about it, it makes sense. It makes sense to turn off the enzyme to stop making tryptophan, and it makes just as much sense to stop making enzymes that are helping make tryptophan. So you need to be able to turn off both. You want to stop the enzymes from functioning, and you also want to stop the production of those enzymes when the cell no longer needs it. So that's what the difference is. And our focus really in the next couple of minutes is going to be about what's going on here. So if you want to go back and revisit feedback inhibition, I believe that's chapter 5, I mentioned it. But feedback inhibition is talking about you stop enzymes that are already made. Gene expression, if we inhibit it, we're stopping the production of the enzymes altogether. So when we think about bacteria metabolism, again, metabolism is talking about a sequence of reactions. These bacteria cells, they're so simple and so small, they have to be very, very resourceful. So that's going to be our focus, right? So gene to protein control, like I said, our focus is on gene expression. Genes are only going to be expressed when needed. So it makes sense. These bacteria cells are only going to make enzymes or proteins when needed. And so it's often regulated or controlled at the transcription level. So remember, transcription is the making of mRNA. So we're going to stop making mRNA. It's going to stop making the protein. Okay. So let's kind of get into it. Here's kind of the key players and what we need to know. We got promoters, operators, repressors, regulatory gene, and structural genes. So before we dive really deep into this, let's at least remind us what is an operon. Okay. It's a group of genes that are transcribed together, and they code for similarly functional proteins, meaning they're all going to be associated with doing the same job. That's what operons are, okay? Now, with that, let's talk about an operon. And before I do that, let me just kind of emphasize one thing, right? So if we had a little bacteria cell, right, you have these big circular DNA, right? You have little tiny circles of DNA called plasmids. A plasmid can have multiple operons. So you got to understand that. An operon is just a region of DNA. I could have an operon here. I could have an operon here. I could have operon here, here. So operons are going to be found all over a bacteria's genome, right? So this is showing you an operon. We have this gene. We have this sequence of genes as well. So what is an operon? First and foremost, they're genes grouped together with related function, okay? All enzymes in a metabolic pathway, meaning they're all going to be associated with the same end goal. So if we jump back to this, this first one, all of these enzymes, 1, 2, 3, 4, 5, all these enzymes are associated with helping to make an end product of tryptophan. So that's when we say that they're all related in the same metabolic pathway. So let's talk about what's going on here. The first ones we got are going to be considered a promoter region, okay? Just like in chapter 17, RNA polymerase, which is this guy right here, I'll kind of outline it for us. This is your RNA polymerase right here, that I'm shading in purple. The promoter is the region of DNA where RNA polymerase binds, okay? Now, we have an operator, which is the DNA binding site of a repressor protein, okay? Now, how do these genes get turned off or turned on? It all has to stem with these repressor proteins, okay? So I'm going to add a few things here. This is going to be what's called a regulatory gene. So let me kind of jump back and forth, okay? So the regulatory gene produces the repressor protein. So this thing right here, before all of this operon, this is going to be called your regulatory gene. And what it's going to do is it's actually going to make a repressor protein. So let me kind of show you how that looks like. So the repressor protein is going to be a protein that can bind to the operator, okay? So this is your regulatory gene. The regulatory gene makes a repressor protein, and then it all just becomes a matter of, well, how does this repressor protein get turned on or turned off, okay? So let me kind of show you this visual. Actually, I'll stick out the tryptophan visual right here. So if you look through this, this is a regulatory gene. It's a sequence of DNA that makes a tiny protein. Now, hopefully you can see right here that when we're looking at this repressor protein in its current form will not be able to fit to this region. This region right here is called the operator, okay? So think of the operator as like the on-off switch, okay? The repressor protein needs to bind here. This thing is going to be the enzyme RNA polymerase. So as it sits now, without this tryptophan molecule, right, This tryptophan binds to the repressor protein. It changes the shape, and now this repressor is active. And then it's going to bind here. And it's not shown here, but let me just add it. What this DNA is going to be having here, DNA that's after this, is going to be genes that make tryptophan. So let's think about it from this standpoint. Let me jump back here. If this first example is about how do we turn off gene expression, right? Well, what if the cell made a bunch of tryptophan? Well, that tryptophan is going to bind to one of those repressor proteins, and it's just basically letting the cell know, hey, we have plenty of tryptophan. We don't need to make any more enzymes to make tryptophan. And that's what this whole model is showing you. So you have the regulatory gene. So this would be called the regulatory gene. The regulatory gene makes the repressor protein. Without tryptophan, the repressor protein isn't able to bind. So then that makes sense, because if you're trying to read these genes to make tryptophan, it makes sense that you want to be able to read this sequence of DNA. Now, if all of a sudden a bunch of tryptophan is there, tryptophan binds the repressor, it changes shape, and now it blocks it. So let me just kind of show you this model of a trip opera, okay? So let's say all of these genes here are associated with making enzymes to help make tryptophan. So think of tryptophan as your end product, okay? So as it sits right now, RNA polymerase can read these genes, gene 1, gene 2, gene 3, gene 4. I'm doing transcription and translation, right? All of these genes are making the enzymes, and all of these enzymes are responsible to help making tryptophan, okay? So as long as I'm reading these genes, I'm making enzymes, and I will eventually make a bunch of tryptophan, okay? So all of these enzymes are involved in making tryptophan. And watch what happens here. The more tryptophan I have, tryptophan can bind to the repressor, and then this is associated as an active repressor protein. So look what happens here. This is now blocking RNA polymerase. So as RNA polymerase tries to come through and read these genes, it can't, because it is blocked by the repressor protein. So that's an example of a repressible operon, okay? So in terms of this, by adding tryptophan, you made this repressor active, okay? That's why we call it a repressible operon, okay? My hint is, well, I'll tell you in a few minutes, but that's what an operon is. It stops the expression of it, right? So in terms of this, watch what happens. Here's tryptophan. If tryptophan is absent, the repressor is inactive, and the operon, or all these genes, are turned on, okay? So basically what this shows is we've got the TRPE, the TRPD, all of these genes make enzymes for tryptophan synthesis. So we got the regulatory gene. This, again, is your regulatory gene. This makes your little repressor protein. Right now, your repressor protein would not be able to bind here. And that makes sense because there's no tryptophan. So what three things make up an operon? You got the promoter, the genes of the operon, and the regulatory gene, right? Those are the big three. So let's say all of a sudden you make a bunch of tryptophan. Where is that tryptophan going to go? Well, it's going to go bind to the repressor protein and make that repressor active, and then it's going to stop the synthesis of all this, okay? So what happens when tryptophan is present? Binds to the repressor, and it turns off the whole pathway, okay? So, and it makes sense because you don't want to make enzymes to make tryptophan. So that's one model of an operon, okay? That's called a repressible operon, because adding in lactose represses protein synthesis. So let's look at this one. I want to say that this next one is talking about tryptophan. Yeah, this one's talking about the lac operon. So let's take a look at this one. So let's think of how to break down lactose, right? So we got a regulatory gene, we have the lac operon, and try to associate the difference between this one and the previous model, okay? So all these are going to make enzymes to help break down lactose. So if you're familiar with lactose, lactose is the sugar you get from things like milk. You need the bacteria to help break down your lactose. So let's see what's going on here. You got a regulatory gene, right? There's nothing there. So I have RNA polymerase. I can make all these genes. And so these are the lac operon mRNA. So what's going to happen is it's going to be basically expressed because there's nothing blocking, okay? Now, check out what's happening here, though. Look at the difference between this visual and the last one. My regulatory protein, or my repressor protein, right, is stuck already, okay? So think of it like this. In this case, I need to get this repressor removed. This is an inducible operon, and hopefully you understand why. How do I get this thing removed? Well, watch what happens here, okay? Regulatory gene in this model makes a repressor protein, and that repressor protein immediately binds to the operator, okay? So understand what's going on here. How is this different? Well, remember in the trip operant, that tryptophan wouldn't bind there unless it had tryptophan bound to it, okay? But notice this one, I didn't have anything bound to it. And again, this is a totally different model. Don't get this confused with the trip operant. All of these enzymes are going to help make breaking down lactose possible. So all these enzymes are associated with breaking down lactose. So let's think about it from this standpoint. It would not make sense for me to make enzymes to break down lactose if lactose is not there in the first place. So let's see what happens here. If I let this thing play out, if this is essentially lactose, right, in terms of this, lactose doesn't bind there. So that lets this cell know, okay, it's time to make some enzymes to help break down lactose because I have lactose here. So that's kind of the whole notion of what's happening with this lac operant. I'm going to help make it. I'm reading these genes, and let's see what these genes are going to do. Each one of these genes are going to be breaking down, making enzymes to help break down lactose. Okay. So if we let this thing continue on, this model will kind of clarify everything up, right? So if we look through this, this is the enzyme to help break down lactose, right? So that's the enzyme breaking down lactose, enzyme breaking down lactose. All these genes made that enzyme to help break down lactose, okay? Now, as all of the lactose has been broken down, notice that your repressor protein can bind back. So check out the difference between that versus the tryptophan operon. There are quite a few similarities, but looking here, the repressor protein is active, so RNA polymerase can't bind there. So I'm not going to read these genes, but notice what happens. If I added in a bunch of lactose, lactose actually removes the repressor. Now I can actually express and read the genes to break down lactose. So RNA polymerase is now going to be expressed. I should say each one of these genes is now going to be read. I'm now going to make the enzymes to break down lactose. Okay, and that's called an inducible operon because by adding in lactose, you are inducing or turning on protein synthesis. So what happens when lactose is present? We'll see here. Here's your repressor protein. Without lactose, that repressor is going to fit right on the operant, or excuse me, the operator. Only when there is that inducer, in this case it's lactose, it pulls it off, you can express the genes. It makes sense. You only want to make enzymes to break down lactose if lactose is available. So in terms of that, how to tell the difference between a repressible versus inducible? A repressible operon is usually on but can be inhibited, right? So think of the trip operon. Tryptophan actually shuts down the production, okay? When the cell has made too much tryptophan, it stops it. Inducible means it's usually off, meaning that whole metabolic pathway, reading those genes is usually off, but it's only turned on when an inducer removes the repressor. In that case, lactose removed it. So typically if it's anabolic, if you're building something like tryptophan, when you're building it, anabolic pathways usually have repressible operons, inducible ones usually have catabolic ones, okay? So your biggest challenge is going to be, can you identify inducible versus repressible operon? So let's do a quick overview, and this one's from your POGL. I like it a lot. So in this case, you have your regulatory gene, you make a repressor protein, it can bind to the operator, okay? Now notice when you added an inducer, you pulled it off, you can express these genes. So by adding in a molecule, you are actually inducing protein synthesis. So I would call this one an inducible operon, because look at what was added. When you added this molecule to your repressor, it pulled it off of the operator that allowed these genes X, Y, and Z to get expressed. Now, on the contrary, look at the difference here. In this case, your regulatory gene, without anything bound to it, it can't fit. It will not be able to fit to the operator. But once all these proteins basically build up and make your end product, let's say this is tryptophan, it binds to the repressor and it makes it active. So basically, in this case, your end product shuts down the production of those enzymes. So this would be a repressible operon, okay? Yeah, this kind of overviews a couple of key things, right? So just to kind of remind you, it makes sense that if this was the lac operon, right? If you had lactose, you had all these enzymes to break it down. This is basically lactose. It pulls off your repressor. And that's how these little cells can control protein synthesis. So a nice little compare contrast on inducible versus repressible one. They both function in protein synthesis. They both have promoters. They both have operators, repressor proteins, regulatory genes, and structural genes. But if it's inducible, remember the repressor protein is made. The repressor protein binds to the operator. The inducer, things like lactose, it's going to remove that repressor protein, and then you actually express it. If it's a repressible operon, okay, the product binds to the repressor, and then it stops protein synthesis. So that is gene expression in prokaryotic cells, and we will see you guys in the next video.